Thank you. If you were here this morning, you heard a lot of examples of knowledge networks, knowledge graphs that include genes and proteins, diseases and drugs. My group is really interested in understanding cellular phenotypes, and we work on the development of a lot of methods to understand single cell transcriptomic data and other kinds of single cell data sets. And so what we wanted to be able to do is to try to add in information about cellular characteristics into these types of knowledge graphs that also includes genes, proteins, diseases, and drugs to try to get a sense for what are the cellular contributions to disease pathogenesis. So I'm at the National Library of Medicine in the United States. We develop a lot of data and knowledge resources. I'm sure this crowd knows about most of them, so I'm not going to go through that. But I think one of the things that we really try to focus on is develop trusted sources of information and knowledge. And that's going to be a theme I'm really going to try to emphasize in my presentation. We have NLM and also EBI and other groups have developed data resources and information resources like GenBank and Geo or RefSeq and Kebi and Kemble. But our knowledge that's derived from the interpretation of those data and information is buried in the scientific literature. And so there is this gap, I think, between our databases and our knowledge resources. And so what we would like to be able to do is leverage all of these resources and develop computable forms of knowledge in the forms of knowledge graphs. And I think many of the people who spoke earlier this morning, have this similar objective. So I mentioned that we're very interested in understanding cellular characteristics and developing a knowledge resource that captures that kind of information, mainly emerging from the single cell technologies that are really kind of revolutionizing our understanding of the cellular complexity of tissues. And so if we can build that kind of a knowledge base, then that should allow us to understand the cellular contributions to disease pathogenesis. That could also help us identify diagnostic biomarkers and potential therapeutic targets. And it turns out we actually know very little about the cellular contributions to disease pathogenesis. But I think now we're at a stage where we can really start to address that. So we want to focus on trusted knowledge sources. So the knowledge base, the knowledge network is being populated using validated data processing and analysis pipelines that can process the single cell omics data. We have reference ontologies that you've heard a lot about throughout this meeting. That's a really rich source of knowledge that we also want to incorporate. We have hopefully using large language models and other approaches to do text mining, we can extract similar types of knowledge from the scientific literature. So we're hoping to be able to take a data-driven approach to take data and translate it into knowledge, and then take a text mining approach to take text and translate that into computable forms of knowledge. And these efforts are really designed to be synergistic with other kinds of community resources like Cell by Gene and HubMap, which you may be familiar with. And we think that there is this interesting niche of representing the knowledge from these kinds of experiments. So I think the key characteristics for this pilot that we're developing, first and foremost, we want the content to be trustworthy. And that includes applying quality control, tracking provenance of the information that's incorporated, focusing on knowledge that's reproducible, and also providing statistical or quantitative evidence for assertions that we're making wherever possible. We need the knowledge base to be evolvable and expand with new knowledge as new experiments are being done. And we want it to be networked with other trusted sources of knowledge that I'll talk about. And it should also be use case driven. And one of the main use cases that we want to be able to address is what cells contribute to the pathology of disease X. It's a semantic network, so it's built on obofoundry ontologies and other semantic principles. It's designed to be fair by utilizing open data standards like BioLink and LinkML. We want to make it AI ready so that the content of the knowledge base can be used to drive model development and testing. And as I mentioned, this is really focused on understanding cell characteristics. So there's two aspects of this that I just want to emphasize during my talk today. One is this question of trustworthiness, and the second is an example of this kind of use case. So here I have the all of the semantic triples that form the foundation of the knowledge base. So we want to utilize standardized semantically structured assertions of subject, predicate, and objects, where biological entities are nodes and the relationships are the edges in the knowledge graph. And all of this is represented as linked open data using BioLink and LinkML. And these are just a few examples that I've highlighted in the red boxes. So for example, we have cell type subclass of cell type. Those are assertions that come from the cell ontology. We have drug product molecularly interacts with a protein. That information is coming from Kemble. We have gene is genetic basis for condition. That information is coming from a variety of different sources that show kind of genetic phenotype interactions. And then we also have gene part of biomarker combination. And I have my favorite definition of what a biomarker is, and we could talk about that over wine maybe tonight. But that particular relationship comes from these data processing pipelines. So then if you link all of these kind of triple assertions together, you get your standard network, and this forms the foundation of the graph that we're populating. So this is, if you come to this kind of prototype, this is the user interface. You can browse, you can search in different ways. So here's one example of I'm looking for information about lung parasites. I can enter that as a search term. And then here is the initial graph that you get from that search. And so over on the right-hand side, you can see, for example, that lung parasite is part of a pulmonary alveolus, and it's also part of lung. It selectively expresses these marker genes that are very specific for lung parasites in the lung. And so these are the kinds of nodes and edges that you would get in this kind of initial graph that's generated. And then on the left-hand side, we have information about this cell type. And in this case, this information is coming from the cell ontology. You can also kind of browse the collection of entities that are represented. So in this case, if you click on the cell type collection, these are the different cell types that you can find in the knowledge graph. And this makes it very easy for you to kind of launch into the graph in specific places. So now in terms of the trustworthiness, the first one is tracking provenance. So we're capturing all of these triple assertions. Where are they coming from? So on the left-hand side here, we just have kind of a vanilla graph that is just labeling the nodes in the graph. But we have the ability to adjust the visualization and add in information about which collections are the nodes coming from, what's the source of the assertions that are being represented in the graph, and also what type of edge is connecting the different nodes. And so that's what you see over on the right-hand side, and then I've just kind of expanded that here. So here you can see that we have on the left-hand side, these are all anatomical structures that are labeled with Uberon terms and identifiers. The lung parasite is a part of those anatomical structures. And that information is coming from HubMap, so that's the source of the information. We have genes that are part of marker combinations, and that marker combination is derived from an algorithm called NSForest that we've developed to identify biomarkers for cell types. We have Mondo terms for diseases, and we have this relationship of genetic basis for disease, so that gene is a genetic basis for that disease, and that information is coming from open targets. And then we have gene produces protein, and the proteins molecularly interact with those chemical compounds from KEBI. So in this way, you really know where all this information is coming from. We track the provenance for every assertion that we're making in the knowledge graph. And then in terms of quality, what we're trying to do is we're trying to develop a really kind of a high throughput pipeline to take data sets from single cell transcriptomic experiments, process them through standardized validated analytical workflows, and we're using the Nextflow platform in order to really scale that analysis. But there are thousands of single cell RNA-seq data sets in repositories like Cell by Gene, and I can tell you the quality is kind of all over the map, right? So is there some way that we can apply some quality statistic to select which data sets we want to load into this trustworthy knowledge graph, and even for some specific cell types, whether there's sufficient evidence to support, their existence? And so what we decided to do is utilize what's called a silhouette score, which captures kind of the tightness of clusters in these transcriptome embedding spaces. So on the left-hand side, you see that yellow snake. That's not a very tight cluster. It's kind of all over the place. Suggests that there's not a really tight transcriptional profile that's represented by that group of cells. And so if you run that data set through our Nextflow pipeline and you calculate silhouette scores, you can see that the scores range from -1 to 1, and most of the cell type clusters have a value less than 1. And so that's not very, or sorry, less than 0. So that's not very high quality, and that kind of reflects what we see in the UMAP embeddings. And so this is a data set that we're probably not going to bring into the knowledge base. On the right-hand side, there's a data set that's much better, and you see that the silhouette scores, many of them for the different cell type clusters, are close to 1. So this seems to be a very high-quality data set that we would want to bring into the knowledge graph. However, there are a few cell types over on the right where you have relatively low silhouette scores. We're going to load that information into the knowledge base, but we're going to keep track of those scores so that you could then filter the content of the knowledge base based on this quantitative assessment of quality. And so in the far right, those are a mast cell cluster. And if you look at the UMAP embedding, those are the kind of the green dots, and you can see that they're actually segregated in 2 separate spots of the UMAP embedding, and there's some scattered all over the place. So I would not rely on any kind of characteristic for that cell type because I'm not sure it's a discrete, entity. So another place where we are capturing quantitative evidence is this NS-Forest algorithm that is able to quantify and identify biomarkers and quantify their performance characteristics. We calculate an F-beta score. In this case, this biomarker combination can identify lung parasites with an F-beta score of 0.84, which is pretty good. So we capture that information here. And again, you can use these quantitative values of evidence in order to filter the graph. I should also point out that all of the analysis that was done to produce those biomarker combinations and scores, we have submitted to Zenodo. And so there is a link to the actual workflow, the data that was used to generate, this information. And so another kind of feature for trustworthiness is that we want things to be reproducible. So now here's an example of part of the graph where we have this. These are the different cell types that you find in lung. This information is coming from 2 Atlas publications, Sycama and Guo, which are in the kind of the mustard colored circles. They link to a data set. That data set identified all of these kind of cell types in the blue circles. But the ones that are in the middle, those are the same cell types that were identified in these 2 different Atlas publications. And so now here, this is an example of how we're reproducibly able to identify cell types from multiple experiments and multiple publications. And so then we can kind of expand that out a little bit. And so we've done that for one of the cell types here. This is lung parasite. And now we can dive in and see some of the additional details here. And so we have things like there's lung parasite. There are 2 marker genes that are very specific for that cell type. One of those marker genes is a potassium channel. So the CNK3 produces this potassium channel. That potassium channel molecularly interacts with those yellow drugs, and that gene is a genetic basis for pulmonary hypertension. So this, first of all, what this does is it actually connects pulmonary hypertension to the cell that we think is playing a key role in that disease, lung parasites. And that's not too surprising because over in the right-hand diagram over there, you see that lung parasites actually wrap around capillaries. And so you can imagine if there's some abnormality in a lung parasite, that could result in hypertension in the lungs. But the other thing is that now here's a hypothesis. We have these drugs that target that potassium channel. They're potentially therapeutic candidates for pulmonary hypertension because of these relationships. And so what we're realizing is that what we want to do is look for reproducible topological structures in these knowledge graphs. And so this is something that we're calling a semantic Big Dipper. And this structure is very interesting because you have a cell type that expresses a very specific marker gene. That marker gene produces a protein. The protein is a target for a drug. The marker gene also predisposes to a disease, and the drug can treat the disease. So these kinds of structures, I think, are very interesting to come out of the knowledge graph. And this allows us to kind of connect cell types to diseases. But the example that I just showed, there's one of the edges is missing. We don't have this edge between diseases and drug products. So this is a hypothesis now that we can go ahead and test. And so what we want to do is develop kind of query algorithms to search the knowledge graph to look for these kinds of recurrent topological structures for things like drug repurposing. So this is what we're doing. We're developing these kind of validated computational pipelines that can take data, populate that into knowledge graph structures, integrate that with other trusted sources of knowledge like ontologies and other databases. And then also we're also applying natural language processing and text mining to try to extract similar information from the literature. And if you're interested in that project, Noam Rotenberg, who's in our group, is going to be presenting tomorrow in the text mining track. So I'll just stop by thanking a lot of people who've been involved in this project, including some people in the audience like Chris, and especially the people in my group, Renee Zhang, who is my statistical kind of colleague in crime, and a lot of the people in the group, Bing Feng, Ajit, Noam, and Matt, who have really been instrumental in developing this resource. So with that, I'll stop. Thank you.
So we have time for a few questions.
Yeah, great talk. So my question is about what was on slide 14, which is about the silhouette scores.
Yeah.
If you're going to say for one data set, this is just so bad, we're not going to include it. But for another one, overall, this is pretty good, but there's some bad stuff, and we're going to flag those. Why not just include the bad stuff with a flag as well in case someone needs it at some point?
Yeah, we could do that, and I think we're going to keep the bar very low in terms of what data sets we would, not include just for that reason. But I can tell you that any silhouette score threshold that I would use, I would get very few cell types from some of these data sets that are just so low. But I think that's a fair point.
So Richard, I think it's great that NLM is producing stuff like this. Thank you very much for pushing forward on all these things that, some of us have been advocating for forever.
Right.
I have a couple of questions about trustworthiness.
Yeah.
And so there are traditional problems that we have with building these knowledge graphs. And so one of them is the logical consistency thing. And I'm going to channel Rob for a second. So did you do any kind of inference, description logic tools or the like to try and find logical inconsistencies in the structures that you built? Mm-hmm. And if you did do that, what did you do about quantification? So that's often where these problems come from.
Yeah. We haven't really done an extensive evaluation of that. That's clearly something that we need to do. This is really a prototype. We're just trying to get all the pieces put together and making sure that things are working. But I think that's something
entirely
that
fair.
Is high priority to do.
second kind of thing around trustworthiness is this stuff changes through the lifespan, and your selection of parasites is a really interesting place to look at that, because they play a critical role in lung morphogenesis, and then at the end of your life they play a pretty big role in idiopathic fibrosis and all these
Right.
kind of nasty degenerative kind of diseases.
Yeah.
So how do you capture and by the way, gene expression profiles change profoundly during that evolution from, before birth. There's a big change at birth, and then the aging process. So how do you handle changes through the lifespan? How do you represent, sort of when?
Yeah, I think what we need to be thinking about is there's all this metadata around these experiments that we're incorporating. And for example, the age of the participants that have, provided specimens. Right now we are not capturing that in the knowledge graph, but these are the kind of layers that I think we do need to add in. Now, how do you capture this kind of temporal change over time? Not sure. I think that's going to be a little bit of a challenge in how we represent that kind of knowledge in this representation.
Okay.
Lots of challenges.
I have one last question.
Thank you. It's a brilliant talk. I'm wondering if you face any challenges in terms of mapping identifiers across this vast amount of databases, such as some single cell data that are mapped to different versions of the human genome. Some only have gene names that got deleted or changed in another version. And what's your plan on keeping these things in control for the trustworthiness?
Yeah. We've definitely had challenges in mapping to the cell ontology, which is kind of our reference of cells, cell types and cell names. And what we've done is, when we're pretty confident that there's an exact match to a term in the cell ontology, we label it as an exact match. When we're not sure, we use a related match or something like that. So we still try to match it to something, but then we also keep track of whether we think it's an exact match or related match.
Okay. So
right.
thank
Thank
you
you.
very much for